So today we're going to talk about some of the interesting work we've been doing at UBC. I remember talking about the concepts that we were chasing in this project probably about 3-4 years ago at one of these meetings. And every year it's crept a little bit further forward, and finally we actually have some what I consider really exciting results to talk about. So let's see if we can make this work. Good. First of all, you'll have to excuse my 69 format. I didn't read the instructions very well. But the rationale for doing a lot of the work that I've been working on at UBC over the last 5 years was trying to solve the problem of how do we find mineral deposits through exotic cover. You look at a map of Canada, and you can very rapidly see that most of the known mineral deposits are actually sitting in areas of outcrop, and there are large swaths of covered terrain in some prime districts of Canada that don't have actually a great deal of mineral deposits on them. In all probability they're there, but they're buried and we just don't see them. So how would we find them? The holy grail of many other research programs. So what we're going to talk about today is the application of using genomics to actually find mineral deposits through transported overburden. And we're going to use in this presentation two Kimberlites that we did a lot of work over the top of, the DI18 Kimberlite and the Kelvin Kimberlite both in the Northwest Territories. Access to both of these was through fantastic cooperation of the Northern Territories Geological Survey, who provided us helicopter support and the whole works that as a university we would never afford. Good. So if we look at how we normally find kimberlites, typically it starts off following an indicator mineral trail, and then somebody wanders around doing a bit of geophysics, and then finally the only option to actually decide whether it is or isn't a kimberlite is to stick a hole down through it, and that's before you even know anything about the economics really. You've got to stick a hole into it to see if it actually is a kimberlite. The object that we were trying to do was can we make that decision-making path after we've done the geophysics to drilling a little easier? Can we provide that additional layer of information that would allow us to make a prioritization decision on which of those numerous geophysical targets, because geophysicists always give you lots of targets, on which of those targets we were actually going to drill. So we're going to start off looking with the work we've been doing at the DI18 Kimberlite. A lot of the base work for this was done by Eureka somewhere in the audience today. She collected all of the samples, including the samples we did for the genomics work. Brilliant, and yes, Eureka did all of this. She was a very busy girl. And in fact, it was one of the great things in the research program that we had was that we started off with a lot of baseline studies, doing surface mapping, trace element analyses, all sorts of other exciting things. And then we could build the projects up as we went along there to add new layers of research onto pre-existing layers rather than starting completely new random projects. If we look at the DI18 Kimberlite, it sits here, as I said, in the Northwest Territories. It's just to the north of the DI27 Kimberlite. That's completely concealed under a lake, as many kimberlites are in the Northwest Territories. DI18 is quite fascinating as it has no outcrop. It sits between 20 to 50 meters of till, and it's actually bisected by a slope break. The northern part of the area is essentially what we would call residual till. It's a till blanket that's been laid down, and apart from stirring and mixing by frost boils, as essentially the same material that the glacier dropped, whereas to the south of that topographic break, you're in an area of considerably reworked materials by fluvio-glacial processes. Eureka mapped all of that out and did all of this beautiful geochemistry, and it was exactly as we would always hope for. We have a nice anomaly in normal soil samples, minus 180 micron upper B horizon, which starts directly on top of the kimberlite and then moves in the down ice direction of the kimberlite, and that's before any indicator minerals come to surface. We're already seeing trace element geochemistry, and as you'll notice if you're looking at those values values, this is extremely low concentrations. I mean, we're looking at a 9 ppm niobium anomaly against a 5 ppm background, but it's really clear. The great thing about it is we could see every single one of those elements, likewise with the field portable XRF. So we'd already pushed forward the fact that we could make a better decision-making process just by using very simple geochemical tools in the field. You'll notice to the south of there, in the area of reworked material, the geochemistry is pretty dead, and this highlights the benefit of actually doing proper ground mapping before you actually go out and even collect samples, so you can make better decisions. One of the other things we did while we was there was we sent all the samples off to another laboratory. I'm not going to mention the laboratory because I'm not doing no product placement. We sent the samples off to another laboratory. They did all the analysis of various hydrocarbons in these soils. And lo and behold, we actually got some really interesting results back, not in terms of the interpretation that they provided us with, which wasn't a great deal of help, but in terms of our own interpretation of the information. We saw all these hydrocarbons that were popping up in exactly the same locations that we'd see in the trace element chemistry. Now, that was really fascinating because it was the first time I'd ever convincingly seen a link between hydrocarbon chemistry and between inorganic chemistry in soil samples. They were all sitting in the same places. I'm very much a process-oriented man, and I wanted to know what was the link between these two processes. One of the obvious links is to look at microbiology. Why? Because bugs produce hydrocarbon compounds as part of their metabolic processes, and when they die, their decay products are nothing more than this whole range of hydrocarbons. When we start looking at bacteria and microbiology, a spoonful of sugar, for example, contains anywhere between 100 million to 1,000 million bacteria. So, sorry, not a spoonful of sugar, a spoonful of soil. Good one. I'm pre-thinking my own punchline there and making a mess of the joke. You certainly wouldn't want to take that after your medicine, that's for sure. Which means we have a phenomenal number of bacterial species in a sample, and since each bacteria contains around 5,000 genes, you can imagine the amount of data that we can generate from this and plow through is pretty phenomenal. But the computing power is there these days, so it's an easy thing to do. And then the other really important factor was the fact that because of the medical sciences, the cost of doing genomic analysis has absolutely plummeted. So the baseline analysis of one of these samples is now less than 10 cents a sample. It really is nothing. The expensive part is doing the processing of the data, but the intention is that nobody would ever have to process the data. We would actually pinpoint the genetic signatures or the genes that they need to look for, and they would only actually look for those within a sample. What can we do with genomics? Two different things. The one is to look at the what's known as the 16S RNA, which is a small section in the RNA that doesn't mutate along with the rest of the RNA and the DNA as bacterial species mutate to make new forms. And so we can use that to identify who was actually living in the soil and how much of them is living in the soil. The other exciting part is to look at the DNA that we haven't even touched yet. And there we can actually look at why are they living in the soil? What are they doing? What is triggering them to make them live in the soil quite happily? Okay, so most of the work we've done so far has been looking at the 16S RNA. And we analysed all of these samples. We were primarily interested to start off with looking at the material sitting in the residual till blanket, because that was going to be relatively undisturbed. But we did include the first line of samples that are sitting off that, so they're actually sitting in the alluvial, fluvio-glucial type of materials, just out of curiosity. Brilliant. So this is genomics quality control. It's a new type of graph for Linda to produce to her collection. What they do here is they keep doing, they continuously take reads of the RNA and just keep adding them together. And then they're counting the number of species that they're identifying. And the idea is that as soon as those lines start to flat out, it tells you that it doesn't matter how much more RNA you carry on reading, you're not actually going to identify any new species. So you've essentially accumulated everything that there is in the sample. The lady who was doing the work, Rachel Simister, our genomicist who works with Sean Crow over at the Life Sciences Department, she stuck this graph on the board and she was blah, 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 bringing a lot about quality control and everything else. And I was completely gobsmacked because those little things on the right-hand side, down ice and above kimberlite, they weren't labeled on her diagram. What was labeled on her diagram was these red lines and blue lines, and it was very immediately obvious that a quality control diagram was actually telling me what I already wanted to see. Something was changing directly over the kimberlite. And we turned a diagram into a simple box diagram using a well-known software package that I'm not going to mention. And very rapidly we could see that above the kimberlite there is a distinct change. We turned that into number crunching, as we always do, and we flip the axis upside down, so we're looking at decreased diversity, right? So the big pink dots are decreased diversity. We're looking at a suppression of the number of bacterial species that are actively living above the kimberlite. Something 20 meters below this till surface in the kimberlite is enhancing, or is having significant effect that it can change the population of bacteria on the surface. Now that was pretty dramatic because for our geneticists, that was the first time we'd had solid concrete evidence that geology at great depth could actually influence something that was happening on the surface quite a way away. So it was really exciting. Those dots that are floating off onto the left-hand side, they're sitting at some of our other geochemical anomalies, so I'm not so worried about those. You can do a lot of exciting things with bacterial species, so you can start to identify which species actually like living above a kimberlite, because there are some species that have preference. We identified at the moment 14 different species that would preferentially live above the kimberlite in contrast to living above the granitic material that's surrounding them. This is some of those 14 indicator species. And likewise, you can do a very similar thing. The diagram is not so great anymore, but showing the same sort of response. We can actually say, well, who doesn't like living over the kimberlite? And likewise, we can pull out a whole range of species that don't like living over the kimberlite. So this was all pretty exciting, because we'd essentially pulled out 3 new layers of information that we could add to our data interpretation that comprised who was or what was the level of suppression of bacterial species over the kimberlite, who liked living over it and who didn't like living over it. Okay, that was 3 new layers that we could add. And that was fantastic. It was some of the most exciting eureka-breaking results that I'd seen over the 5 years that I was working at UBC. It's a shame it wasn't in geochemistry. It was in a completely sideways field, but it was still pretty exciting stuff. However, doing it across the top of one kimberlite is not of use to anybody else, because finding one kimberlite when we know the kimberlite's there doesn't help anybody. So we took the whole work across to a second kimberlite, the Kelvin Kimberlite, also in the Northwest Territories. This is a really strange kimberlite, as we'll see in a moment. This isn't your classic kimberlite. DI18 is your straightforward carrot, comes up to the surface as a carrot shape. This one, it comes up and then stops around 150 meters short of the surface and takes a dive off to the south. It's following some sort of lithostructural control that's controlling the direction in which it actually erupted. The kimberlite likewise has no outcrop. It outcrops or subcrops underneath the lake and then dips off to the north. At the back end of the kimberlite, as I said, there's 150 meters of metasedimentary rocks over the top of the kimberlite, plus 5 meters of till sitting on the top of that material. All of these samples were collected by Andy Wickham, who's another one of my students working on till geochemistry. If we look at the simple geochemistry, all nicely normalized and summed by percentiles, etc., the sort of games we play in geochemistry, and we have a nice anomaly that's essentially it's not as well as the one over at DI18, but it's essentially following the down nice directions from what would have been the subcrop of the kimberlite underneath the actual lake. So what did we do? We gave the samples to the geneticist, And we told them not to go and search the whole database again and try and see what exciting things they could find in it, but on the basis of what we'd already seen over at DI18, tell us, are those same things happening above this kimberlite as well? I must say at this point that my hopes were not particularly great, because there's 150 meters of rock sitting over the top of this, and that's an awful lot of material to try and cut anything through. However, much to my surprise, I love Eureka Moments, much to my surprise, we started plotting up these various variables that we'd seen across DO18, and they were coming out just as good across DO27. So decreased diversity, for example, the suppression of species living across the top of the kimberlite, we're seeing exactly the same pattern at the DO18, the Kelvin kimberlite pipe. At the back end of this kimberlite pipe, through 150 meters of rock, that kimberlite is putting enough of some sort of effect on the surface environment that we are suppressing the bacterial species living there. Now that was really exciting, because this was the first time I'd ever seen any convincing example of deeply buried deposits actually having a direct influence on the surface environment. And likewise, if we take those 14 species that we'd previously summed at the DO18 Kimberlite and do exactly the same thing with the same species at Kelvin, we see an anomalous response in exactly the same place over the back of the kimberlite. So that's all really exciting. What we want to do next is to get some more kimberlites and do it, because 2 is good, 3 is not a crowd, 3 is a phenomenal amount of phenomenal amount of success. So what do we have? We have, in conclusions, we have a direct target identification of a kimberlite using microbiology, irrespective of the till medium, so that we can see that influence both over the residual till blanket, as well as through the fluvio-glacial material that is sitting off the, often burying the till blanket, and up to 20 meters of till. We've got some reproducibility of the application between deposits. That's 2 separate kimberlites, both showing the same effect. And perhaps more exciting, we've got direct target identification through bedrock, so we can actually see that kimberlite through a considerable thickness of bedrock as well. Why, you may ask? Okay, so I'm certainly a process-oriented man, so I want to know why. Why are we seeing that sort of influence? As I said, my students all measured every possible thing under the sun that they could measure over the top of these kimberlites. We have all of the surface geology, all of the physicochemical variables, pH, redox, the whole works, all of the trace metal chemistry, subject to the limits of detection limits of the analytical techniques. There are some things that we don't have over the kimberlite, and it's one of those times when you really kick yourself in the back for having not thought ahead and gone out and measured them. We don't have any measurement of things like methane, carbon dioxide, and hydrogen, that are all gases that could be generated by serpentinization processes in the kimberlite as you convert olivine to serpentine and other products. And our feeling at the moment, certainly when you look at 150 meters of rock, is that it is probably gaseous products generated by the weathering of the actual kimberlite that are exerting that influence on the surface. And to that extent, we have a whole series of experiments going off in the laboratory at the moment, where we're just sitting in kimberlite weather and actually measuring the gaseous products coming off of it. The thing that I would like to do next, which unfortunately I'm not going to get to do, would be to fly back up to both of these kimberlites with a gas probe. One of my other students has been using a gas probe that collects light gases, the hydrocarbons and carbon dioxide and hydrogen as well, across the top of mineral deposits, just using a very simple sucking system, and then you send those off for analysis. It would be great to go back again to the Kelvin pipe. That's an easy one to get to. There's an S-strip next to it. DO18 is a little more difficult, as it really is in the middle of nowhere, and actually measure those gas concentrations to see whether we can put a better understanding of what's controlling the changes in that surface environment. And then the next part of that is to refine all of this for kimberlite deposits. Can we define exactly, super clearly, what it is that we should be looking for across the kimberlite in terms of the microbiology? Because if we can, then we can push that forward into a protocol of generating essentially a handheld device that would allow you to identify very specific bacterial species in a soil sample in the field, which of course is the end point of all of these types of exercises. And that is it. I'd like to thank my colleagues. Bianca's the student who's doing all of this work. She's another MSc student who should finish off this year. Sean Crowe is a very lucky man. He's a geochemist, which covers just about every possible field you could ever want. And Rachel Simister is the delightful microbiologist who does all of the DNA work and the number crunching. And then, of course, a whole pile of students who, completely unbeknown to them, when they were doing their geochemical work, they were actually collecting all of the samples that were being stuck into deep freeze so that we could subsequently do all of the microbiological work. And last but not least, no product placement, but thank you to all of my various sponsors who've sponsored me over the last 5 years with money and in-kind support and instrumentation as well. Thank you.